Hello, I am Bhagat Singh and I am a PhD student in the Department of Clinical Neurosciences at the University of Calgary. I work on peripheral nervous system and why this is important. Peripheral nerves are essential connection between neurons, between neuron and muscle and skin. Peripheral nerves connect brain and spinal cord to the rest of the body. And we need these peripheral nerves to walk, to move, and to feel sensation like heat and cold. The sciatic nerve which I am working on is the longest nerve of human body, which runs from the spinal cord to the foot, and it carries thousands of axons. I work on peripheral nerve injuries, and unfortunately, these injuries are very common, and they leave patients with lifelong disabilities where these injuries rob patients with their movement and sensation. As axons have long distances to regenerate or to grow, it's very hard to revive their function, and the regeneration process is very slow, poor, and often incomplete. I work in the laboratory of Dr. Doug Zacardney, who is an expert in the field and a clinical neurologist who can appreciate the struggle of his patients suffering with peripheral nerve injuries. Neurons are electrically excitable and they can communicate with each other by passing electrical signals like a relay runner passes a baton. My project is specifically looking at the role of electrical stimulation on peripheral nerve regeneration. And when I stimulate these neurons, they fire action potential which travel back to the cell body in the dorsal root ganglia or spinal cord, and they ramp up their regeneration machinery. That means they start producing more of proteins or growth factor, which are essential for the nerve regeneration. Using a specialized equipment, in this case, a microelectrode array, this is a beautiful device where I can stimulate either a single neuron or a cohort of neuron isolated from rat. And when I grow these neurons on this chip or a dish, they start extending new process, which is pretty amazing, because after 24 hours after stimulation, I see the new axons are sprouting, and which are essential for nerve regeneration. In animal experiment, after nerve cut, we were able to show that with electrical stimulation, there were a higher number of axons were regenerating, and they were also growing at faster pace. Using a genetically modified mouse, where we can trace fluorescent axons growing in a live animal, we found that the new axons were visible in the toe pad and foot pad of the animals 2 weeks earlier than control. And they were also doing good on behavioral tests. That means their recovery was better. Clinical studies have shown that electrical stimulation brought greater improvement in hand functions in patients with carpal tunnel syndrome. Now we are working on designing a new biodegradable and flexible chip which can be implanted on damaged nerve. My hope is that my research on electrical stimulation will lead to new treatments for patients suffering from peripheral nerve injuries. I can dream of a day when you have injured your leg and lost sensation in that area. My clinical colleagues will be able to implant the biodegradable chip to restore and regenerate your nerve. Thank you for your attention.